Hi, we're Deep Brains, and with the guidance of Dr. Howard Eisenstein, we present a deep learning model to predict the stage of Alzheimer's in patients. Alzheimer's disease, or AD, is a neurological disorder marked by loss of cognitive functioning and behavioral abilities. It is the sixth leading cause of death in the U.S. It usually progresses in 3 stages, ranging from a healthy brain to complete dementia. Given its fatality and how symptoms worsen over time, early AD detection is a crucial area of research. Brain imaging is a common technique in diagnosing AD, namely magnetic resonance imaging, or MRI. In recent years, positron emission tomography, or PET, has been rising as a new option, being able to visualize amyloid beta plaques in the brain. However, PET scans require radiation and are also more complicated and expensive. SUVR from PET scans indicate the level of amyloid content in the brain. This is subsequently used to classify patient status to normal cognition, mild cognitive impairment, or complete dementia. We look to eliminate this added complexity by predicting patient status directly from MRI. Previous studies used simple classification models such as support vector machines and failed to obtain satisfactory results at around 66%. We present Deep Brains, which is a faster and pretty accurate alternative to this. The deep learning model takes MRI images as input and predicts the patient's status, or stage of Alzheimer's, which is indicative of the level of amyloid beta content in the brain. Standardized uptake value ratios, or SUVRs, of the PIB PET images were renormalized to estimate amyloid content in the brain, which was subsequently used to classify patient status, CN, MCI, or AD. These are our ground truth labels. Our model uses T2-weighted MRI images to predict these classes. We hypothesized T2 images to contain the necessary contrast to predict amyloids, and thus patient status. Past studies reflected similar success with T2 images among other MRI modalities. We use the ADNI dataset, which has over 1,800 participants, more than 8,000 PET scans, and more than 20,000 T2-weighted MRI scans. For our dataset, we selected 8 commonly used T2-weighted MRI imaging techniques and aggregated images collected using these methods into 8 separate NumPy arrays. Each of these arrays has a corresponding label array designating the patient status as MCI, AD, or CN. We tried different CNN architectures for this problem, but got the best result with ShuffleNet version 2. The input for the model was 3D-aligned MRI images. The hidden layers in the model can be split into 3 parts: basic blocks, down blocks, and channel shuffle. For the basic block, we used 3 convolutional layers followed by a batch norm layer. Then the output of the basic block is split into a right and then left down block. Finally, the output of the 2 blocks are concatenated, which then is further passed through the channel shuffle in order to allow information flow across feature channels. At last, the convolution layers are followed by an output layer comprising of 4 linear layers with ReLU activation, and classifies the hidden layer output into 3 classes. We use Atom Optimizer for training and the cross-entropy as a loss function. This work is an extension of research done by Professor Eisenstein's students. Their approach led to an accuracy of 66.39%. We trained 6 different deep learning models on our dataset and measured performance based on prediction accuracy. We found that the lighter weight ShuffleNets and mobile nets performed best, while ResNets performed worse. We hypothesized that the lighter models performed better as the reduction parameters made them easier to train. Our results are promising because with an accuracy of 85% with ShuffleNet version 2, we expect that the deep learning models may soon be able to predict patient status with a high enough accuracy to replace more traditional methods of diagnosis. This is an emergent field of research, and there are a few possible directions to take this next. Some possible next steps are adding other proteins like microtubule-associated protein tau to identify SUVR status. Another possible approach is to take voxel-wise SUVR for the regions of interest, instead of averaging the SUVR for the complete MRI image. We could also use more compute power and regularization for the bigger networks. Thank you for watching.